So what I'm going to talk about is metagenomic sequencing, and that's what we were performing at campus today. So when we say metagenomic sequencing, we mean we're sequencing directly from the environment. So for most of the people in the room, the environment is pig samples, so nasal swabs, serum, oral fluids, tissues. And again, I got a couple of columns here talking about Sanger sequencing versus metagenomic next-gen sequencing. So we've heard a lot of this already, but typically when we run metagenomic sequencing at K-State, we generate about 1 million reads per sample. We paired 150 base pairs, so we get 300 base pair reads with a little bit of IC. K-State's charging $300 per sample for metagenomic sequencing. And again, this can be targeted or not targeted. You know, next-gen sequencing, this means we generate a lot of sequence. And there's different ways of generating that sequencing. Most of what we do is non-targeted, so we use random primers. The benefit of this is we can pick up quasi-species, we can pick up multiple different species, and depending upon how much virus is in the sample, we can get complete genomes for the viruses present. So a lot of times we don't have to do virus isolation. We can pull these sequences directly from the material. And we saw some background earlier, but this is the workflow that we follow. We take our sample, and the first step is to treat it with nucleases, so DNase, RNase, and that's to degrade unprotected nucleic acid in the cell. We want to get rid of pig DNA, pig RNA. And we'll also send it to sample, human bacteria. So again, what we have then is we isolate RNA and DNA, so it's enriched viral nucleic acid. We use random primers to reverse transcribe the RNA. We then make cDNA, make it double-stranded, and then we amplify. So at the end of the day, we've generated millions and millions of little PCR products randomly from the sample. We then go to our assembly. We use CLC Genomics for all of our assembly, so it's just commercial software. But typically we do de novo assembly. We tell the computer to stitch these sequences together to make contigs, so just bigger pieces of DNA, and then we analyze them at blast. So we compare them to databases of sequences to see, you know, if I've got 1,000 contigs, well, what does each contig belong to? So we'll find curves, we'll find mycoplasma, we'll find pig, because we enrich for viral sequences, but we still have bacteria, we still have hosts. And then, depending upon what we're interested in, we'll do some additional assemblies. You know, like in this example, if I was interested in PERS, I would then reassemble these sequences to a PERS reference and kick out a PERS genome. So that's the general flow. We can modify this depending upon what questions we're asking. And then this is what the assembly looks like. This was a project we were working on a couple years ago, porcine parainfluenza virus. We had samples from pigs with unexplained respiratory disease. We did blast after we sequenced nasal swabs. We saw hits to parainfluenza virus. We then loaded in a parainfluenza virus reference. That's what this is. It's a reference genome. And then we mapped the reads to that reference. So all these little rainbow lines here are individual reads. And what this is saying is that at each position, how many reads cover that. So, you know, it's variable because, again, we're randomly generating a sequence. But the point I want to make here is, for this sample, we actually had quite a bit of reads. 11 million reads on this sample. 52,000 of them mapped to parainfluenza virus. So, you know, on a percentage of read basis, less than 0.5% of the reads in parainfluenza virus. You may think that's not very good. Less than 0.5% is what I care about. But again, since I have 11 million reads, we have such depth, we still get 361-fold coverage of the genome. So that's really the key to next-gen sequencing is just the depth. We have so much power, we can just say we don't need to know what we're looking for. We can randomly sequence everything and sort through it after the fact. So my talk is going to be really focused on the application of metagenomic sequencing for veterinary diagnostics. So I'm going to give examples of cases that we processed at K-State. You know, my favorite ones are the unusual clinical presentations. Someone calls, pathology calls and says, hey, I got a really weird case, this is what we're seeing. They think it's virus. It's negative on all our PCR panels. So those are the fun ones. Hopefully we're going to find something new. Likewise, cases where, hey, this looks like flu, but it's not flu. We don't know what's going on. We do this for herd profiling. And also looking at feedback. A lot of people expose herds to PED, and they're feeding back PED positive material. But the question is, what else is in there? Or if we're doing serum exposure of PRRS virus, what else are we injecting into pigs besides PRRS? So we do a lot of that testing too. So the first case I'm going to talk about was an unknown neurological disease. It's an outbreak in North Carolina last year. I received a call from the veterinarian. Approximately 700 pigs had died, starting at 5 weeks of age up to 14 weeks of age. Main symptom was intention tremors, so as this progressed, the pigs couldn't walk, pigs couldn't drink, pigs died. 100% mortality in about 4 days. Samples original to Iowa State, nothing of significance was found. Also, several months after this initial outbreak, new pig, same site, same clinical presentation. Oops. So again, 700 pigs at this site died. This is the clinical presentation. So we received fresh brain samples. We did metagenomic sequencing. The only virus identified at 195 reads mapping back to atypical porcine pestivirus. This is a new virus that my group had discovered about 6 months before that. We found this coincidentally as part of the National Pork Board PRRS project. So just like Doug talked about PRRS genome sequencing, we had a Pork Board project to do full genome sequencing of PRRS from serum samples. And as we were assembling PRRS genomes and serum samples, we noticed a lot of reads to showing very low similarity to pestiviruses. So we ended up pulling out a complete very divergent pestivirus genome. They had about 25% identity for BVD, classical swine fever. And what it was most closely related to was a mat pestivirus that was partially described out of China a couple of years ago. When we found that pestivirus, we did go back and make diagnostic assays, including monoclonal antibodies. So the top right here, these are the monoclonal antibodies reacting with, this is cell cultures with marked cells expressing the ERMS protein. You see the green fluorescence. But since we had those reagents available, we were able to go back to pigs' tissues from this outbreak, look at various tissues for pestivirus. You can see staining in the liver, spleen, especially lymph nodes. Also, once we found the virus, we used real-time PCR to confirm it's present in the brain as well as multiple tissues. So, again, we felt pretty confident that this pestivirus is likely a significant contributor to the disease we're seeing out there. Shortly after we found this virus, ISU announced they also were working on this, and they had also found it. However, they found it in baby pigs with congenital tremors. They were able to inoculate sows and reproduce congenital tremors on neonates. So the question we had at the time really is, how serious is this disease? Congenital tremors have been around forever, relatively sporadic disease. However, the presentation that we dealt with, these intention tremors, the high mortality events, I had not heard of previously. So, is this something that's new that we need to be concerned about, or is it something that's been going on sporadically and we just haven't really talked about it? Since we published this work, there's been a lot of work going on with this virus over in Europe. There's been a couple papers come out. I do know that one of the groups over there has shown that this virus, pigs born with this virus, are persistently infected. So again, we're looking at other pestiviruses as models. This virus looks very similar. But again, we haven't known about this virus long, so a lot of research is still being done. For the sequencing work out there, there's a lot of diversity in these viruses, probably upwards of 15 to 20% difference between isolates. So again, what is the effect of the genetic diversity on the clinical picture? And then he also has other questions. Is this a new virus, or has this been there for a long time? Reports of virus X or ST virus-like viruses in the literature for a while without conclusive genetic identification. So again, one application where we use next-gen sequencing. This is another interesting case that we're working on right now. Again, out of North Carolina, sow farm with reproductive problems. We were contacted. They had a bunch of sows that died acutely of lesions of PDNS. Likewise, there were sows on the farm with PDNS-like skin lesions that were aborting. Samples originally went to ISU, and the histopath was consistent with PDNS. However, PCR and IUC for PCV2 were negative. So usually we think of PCV2 as causing PDNS. So at this point, samples came down to K-State for sequencing. We were able to identify analogous circovirus right here. We call it PCV3, so you can see it looks like a normal circovirus. It's a little bit bigger than PCV2, so 2,000 base pair genome. Phylogenetically, you can see it up here. It's more similar to canine circovirus than it is to PCV1 or PCV2. And just a pairwise level, this capsid is only 36% identical to PCV2. So this is a radically different circovirus. We also generated monoclonal antibodies, and we went back and did IHC on the skin, lymph node, and kidney tissues. So we did see nice lesions, typically that we'd see with PDNS. But we also were able to show PCV3 antigen was present in all these tissues. We set up built-in PCR for PCV3. The sows with PDNS had relatively high CT values, upper 20s. However, in the mummies, again, that came from sows with PDNS, we saw CT values 17 to 21. So very high levels of virus. In terms of genome copies, you're talking 7 to 8 logs of virus. We also took a bunch of samples from ISU that were submitted for just unassociated respiratory disease testing. 12% were positive, telling us this virus is out there. We also worked with ISU to go back to their archived tissues, and they pulled 48 cases where they had diagnosed PDNS based on the clinical signs and testing that had tested negative for PCV2. So PDNS cases, PCV2 negative. We tested those by PCR for PCV3, and we found 94% were positive. We also did some confirmatory sequencing on some of these, and then also did IHC, and here they find that we tested by IHC were also positive for PCV3 antigen. So, I think most people know the history of PCV2. First identified in the 90s in Canada, sporadic outbreaks of wasting disease. Wasn't thought it was a big deal until late 1990s, early 2000s, when this really became a worldwide epidemic of wasting disease. And it wasn't until the mid-2000s we had commercial vaccines that PCV2 was controlled. So, of all the viruses that we've found over the years, this is the one I think we should pay attention to, and we need to be proactive about. Really, because it's on a similar trajectory. It's similar to canine circovirus, porcine circovirus. This thing certainly has potential to cause disease. Some of these questions, 12.5% of samples were positive. What's the role of some clinical infection? So that's the PCV3. I have a couple other viruses here we're going to talk about that came from cases sent to K-State, unexplained respiratory disease. And that's a call we get pretty frequently. Hey, we got pigs that are 10 days of age, out to about 4 weeks of age, coughing, their flu negative, their PERS negative. It's consistent. These pigs fall behind. We just can't consistently find influenza or PERS. There's something else going on. So we had a number of those last spring. And we've identified very high titers of a novel astrovirus 4. So it's up here. It's fairly divergent astrovirus. Typically, we find astroviruses in intestinal samples or fecal samples. So this is really unusual for an astrovirus, apparently with respiratory tropism. But again, we consistently find this virus, and again, we published it, and we didn't publish much more than just the sequence and the incidence in the field, so we don't know much about it, but it's one that we see often in these type of testing. So certainly more research is needed on a lot of these viruses. Likewise, on this unexplained respiratory disease, another virus that we find quite frequently is porcine parainfluenza virus 1. This virus was first identified in Hong Kong a couple of years ago. So this was the first identification in the U.S. But again, we had a bunch of 10 to 21 male pigs, nasal swabs that we tested. We were able to find this virus. So you can see the U.S. isolates very similar to these ones in Hong Kong. We did do a little follow-up testing here. We looked at just general diagnostic submissions. We found 6.1% were positive for this virus. We did some serology, showed about half the pigs that we tested were positive for exposure. And we also did in situ hybridization to look to see where is this virus replicating. Interestingly, we only found it in the upper respiratory tract, principally the turbinates and the trachea. For the most part, we couldn't find it in the lungs. So those were, again, kind of client request testing various viruses we find. The last couple examples are kind of herd profiling examples. So this was a summer project with that student, Olivia Myers, North Carolina State. And what they were asking, she's working with girl-to-girl milling. And what they were asking is, in North Carolina, we've got slaughter plants in very close proximity to commercial production facilities. And so they wanted to understand the role of slaughter plants as sites of animal concentration and commingling. And then again, if there are viruses coming in with these pigs, mixing around, could they blow in the wind or trucking to the adjacent commercial sites? So what viruses are present? So we looked at two sites, which were, I guess, primary market sites. So healthy, normal pigs going to slaughter. So these are all the viruses we found. And again, it's not surprising. We find lots of parvoviruses and torpano, the circle virus, a bunch of random viruses. Nothing too exciting on this list. But then we also sampled pigs from secondary slaughterhouses. So mostly cull sows, herniated pigs, fall-behind pigs. And we find a lot of the same viruses. But what's interesting are the ones in the area here. We found a very high percentage of these pigs in Seneca Valley, PEDV, Delta Corona, atypical porcine pestivirus. So some of these popped out. And again, when we found the Seneca Valley, it was interesting. That's about a year and a half ago. So this was right before we started hearing a lot about Seneca Valley outbreaks in the U.S. So we did a little bit more research on Seneca Valley. The genomes are up here, very similar to the Brazilian genomes reported by the U of M last year. But again, what I thought was really interesting, we went back into PCR of these samples. The primary market pigs, only 2.5% were positive for Seneca Valley. The secondary market pigs, 72%. So large discrepancy between the type of pig that we're looking at. We also were able to do virus isolation on these samples. So anyways, it was an unexpected finding and another illustration of when we do metagenomic sequencing, we don't really know what we're going to find. It's great for these cases with unusual clinical disease, but even pigs here, all these pigs are healthy, fit for slaughter. You can still find viruses like Seneca Valley. So really, I think this technique allows you to really investigate the ecology of a lot of these infectious diseases. Another example, we had a grant from the Kansas Bioscience Authority to profile the Virome of feral pigs. We've got feral pigs in Kansas. There's always concern about feral pigs carrying pseudorabies virus, classical swine fever, and who knows what else. And these viruses get into our domestic herds. So the question was, what viruses are in wild pigs? Not a lot of studies. So we sequenced 100 nasal swab pools from wild pigs to see what we could find. To be honest, we found hardly anything. These were the cleanest pigs I've ever seen. Most of the pigs didn't have a virus in them. But we did find one interesting virus, novel orthopneumovirus, right here, swine orthopneumovirus. This was interesting just because there were no swine orthopneumoviruses up to this point. Other viruses in that genus include VRSV, human respiratory syncytial virus, pneumonia virus, mice, and adenovirus. So viruses that cause significant respiratory disease in other species. We did do some serology on both domestic and feral swine samples. We found about 30% were positive. We also did PCR and went back and looked at domestic and feral swine nasal swabs and found about 5% of domestic pigs were positive. So really the question is, what does this virus do? It fits in a family where they all cause respiratory disease. We've got evidence that this virus is in both wild as well as domestic pigs, but apart from that, we have no idea. So again, next-gen sequencing does a great job finding things for us to continue the research. Last couple of slides, a lot of times we find viruses and we've got no idea if they have any clinical significance. Here, the novel parvovirus, we find this in a lot of pig samples. You can see this down here. So it is extremely divergent parvovirus. We know nothing about. Likewise, another group of viruses that we find very often are called posoviruses. These have been found in humans and pigs, lots of species, typically in fecal samples. We published this paper from that the herd profiling experiment I talked about in North Carolina with the slaughterhouse pigs. We found all these complete genomes of posoviruses. So this is in the family Picornaviridae, and within the family, these are various orders. So Picornaviridae. These are orders of coronaviruses. You can see the sheer amount of diversity we find in pig feces of these viruses. And I bring this one up here because I don't have time to go into today, but our hypothesis, and we've got some pretty good results that I think support this, is these viruses we don't believe infect pigs. We actually found these viruses in the water supply at the pig farms. So we think that these viruses infect algae. And essentially, pigs are drinking water, and when they're consuming these viruses, and these viruses are coming up the backside of the pigs. So we did some serology in pigs and found essentially no evidence of infection. Yet we see a lot of these viruses in pigs, and we can find it in the water system of pig barns. So, and that's a good point for metagenomic sequencing. When we sequence, we're going to find pig viruses, we're going to find soybean viruses, because that's what's in the pig's nose. We're going to find aquatic viruses, insect viruses, you name it. So a lot of times, if you find a virus like this, we don't know what the host is. Is this a pig virus? Is this something else? So last slide. Metagenomic sequencing, I hope the message is, this is a really powerful technique. We can use it for a lot of stuff. Viral genome sequencing, investigating the etiology of these unknown clinical cases. It's useful for profiling herds, so again, for live exposure. On a cost basis, K-State is charging $300, and other universities are charging similar amounts. So if you look at running multiple PCRs, histopath, culture, BI, it's pretty easy to get a $300 diagnostic bill. So I think this can certainly not replace all our diagnostics, but certainly has a role in veterinary diagnostics. I'm a big proponent of we need to use this more often. We need more veterinarians to request this. But and the final point here, it's very easy to see we find a lot of stuff that's interesting, but really, more often than not, it raises more questions than provides answers. So be forewarned, you may get an email from Doug or myself or someone that says, hey, we found this and we have no idea what it means. But it's good for graduate students if I'm looking for projects. So finally, just want to acknowledge Virome at K-State that's helped these projects. Collaborated with South Coast State, Iowa State, especially providing a lot of samples, veterinarians for making us aware of these cases, and as well as all the funding agencies that are supporting